In this video, we'll be talking about developmental networks, specifically for the thorny dragon. The developmental network we'll be looking at is that for the spikes created in the skin cells, seen by this network here. This network shows our wild-type condition in which all genes are functional. To determine if spikes are produced, we will follow this network. First, we have S, which promotes P. With this, P is able to function and repress I. Since I is repressed, it is not able to function, which means it will not repress K. This means K is able to function to promote E, which will then promote spikes. So, in the wild-type condition, we produce spikes. Let's look at a few mutations of this network. In this mutation, we have P-, which means P is not functional. Because P is not functional, it will not repress I. This means I is able to function to repress K. With K repressed, E cannot be promoted, which means spikes cannot be promoted, so we will not produce spikes. Next, we have an I- mutation. With a defective I, we do not repress K, which means much like in our wild type, K and E are able to promote and produce spikes. Here we have a mutation I and E. We'll focus on our E mutation because it's further down the network. Because E is mutated, we cannot promote spikes, so spikes will not be produced. Finally, we have a mutated S and K gene. Once again, we'll focus on K. With K dysfunctional, it cannot promote E, which in turn cannot promote spikes, so spikes will not be produced. These mutations supply one explanation as to why some cells may not produce spikes. However, what about in the case of non-mutated cells? We still see some skin cells will not produce spikes. One such example of regulation that may cause this is methylation. Methylation involves the addition of a methyl group to a cytosine. This attracts deacetylases, like HDACs, which causes nucleosomes to pack tighter together and in turn blocks transcription factors from binding. We can visualize this with these penguins. These penguins represent our histones. In their normal state, transcription factors, represented by the puffles, can easily access each histone, allowing genes to be expressed. However, when methylation occurs, our penguins huddle closer together. Because of this huddling, the puffles are unable to get to every penguin, so we'll see that not all genes are being expressed. And that's on developmental networks.